Sup, Science with Chris here. Right now, in the next 3 minutes, we're going to be looking at how to look at proteins of interest in our samples. The first step is to separate them on a gel based on their molecular weight. The heavier ones remain at the top, while the lighter ones race to the bottom. We're adding 1x running buffer to maintain the flow of electricity throughout our gel. I then prepare my samples by heating them for 10 minutes at 90 degrees Celsius. I pipette 20 microliters of sample to each gel lane, and then I add a standard which has proteins of known molecular weight size to compare against my samples. In order to propel my samples through the gel, I connect them to a power source, where I run it at 90 volts until the samples have run all the way to the bottom. After separating the samples with electricity, I then remove the gel and I transfer it over to a nitrocellulose paper, shown in a few seconds. And now I'm beginning the process to transfer the proteins lodged within the gel onto a nitrocellulose paper so they can interact with antibodies. Upon transferring, I then ensure the proteins have transferred to the paper by staining with Ponceau, which looks for proteins. I next wash the Ponceau off and block each of my membranes in a specific blocking buffer. After the blocking buffer, I put the membranes overnight at 4 degrees Celsius in a primary antibody. Next, I put them in a secondary antibody made with 5% milk in TBST. I then finish up by washing the membranes in TBST.